Good morning. Today we will study sporogenesis in Riccia. It is the process leading to the transformation of the diploid protoplast of the sporocyte, that is the spore mother cell, into haploid dispersal unit, the spore with this resistance wall. The process consists of complex integrated series of events involving a special kind of nuclear division called meiosis. Meiosis, the diploid nucleus of the spore mother cell, undergoes successive nuclear division by the chromosomes, but the chromosomes are replicated only once. In Riccia, almost all the cell of the sporogenous tissue differentiated into spore mother cell or the sporocyte. As the spore mother cell begins to separate from one another, they become more or less rounded. Their wall become mucilaginous and the content densely granular. Each sporocyte has a diploid nucleus. With the disintegration of their wall, spore mother cell increase in size and the diploid nucleus undergoes meiosis. The first meiotic division is reductional. It reduces the chromosome number to half and half of the somatic number. The second meiotic division, which follows the first, is equational division. Both constitute meiosis. The resultant four haploid daughter nucleus migrate to the periphery of the sporocyte and lie at equal distance from each other. The cell wall delimiting the four haploid spores are laid simultaneously between the four nucleus at the end of the second division. The group of four cells is called an immature spore tetrad. The cells in the tetrad are typically arranged in a tetrahedral manner, and the wall that separates them is in the form of a thin membrane. The four cells of the immature tetrad are enclosed at first in a common sheath. Each cell of the tetrad, however, secretes around it within its thin membrane a comparatively thick homogeneous callose layer. It is deposited unevenly around the spore protoplast and acts as a mold against which the marking of a mature spore coat are cast. Inside the callose layer is deposited the first or the outer layer of the spore coat, known as the exosporium, is in the form of series of lamellae. The later project into and follow the counter of the callose layer. The second layer of the spore coat, which is known as the mesosporium, is deposited on the inner side of the exosporium. During the concluding stage of the spore development is formed the third and the innermost layer of the spore coat. It is termed as the endosporium. The three layers of the spore coat thus develop in a centripetal succession. As the spore mature, the spore coat thickness considerably. The exosporium, which is yellow at first, changes to orange and become black at maturity. The callus layer and the sheath of the spore mother cell, which enclose the four spores, disappear. Continuously, the spores consequently the spores are separated and lie free in the cavity surrounded by the outer layer of the calyptra. The diploid or the somatic number of chromosome in many species of Riccia crystallina is 16. According to Karasu, the haploid chromosome number in Riccia melanospora and Riccia crustacea is 16. Sporophyte. The sporogonium of Riccia is the simplest among the liverworts. It lacks both the foot and the seta. It is just a spore sac or a capsule, spherical in outline. It has a wall, one cell in thickness. The capsule wall encloses a mass of spore mother cell. The two-layer calyptra form as a close investment around the sporogonium. The latter never emerge out of it. Before the spore mother cell divide to form spores, the single layer wall of the sporogonium disintegrate. Later, the inner layer of the calyptra also break down. The mature spores lie free in a cavity or a sac surrounded by the outer layer of calyptra. There are no elaters. The mature sporogonium of Riccia thus has no diploid or sporopetic structure at this stage. The meiospores, which represent the future gametophyte, are housed in a sac provided by the gametophytic tissue. Nutrition. Unlike other liverworts, the embryo, sporogonium and the spore mother cell in Riccia develop no chloroplast, thus no starch is formed. The sporophyte remain totally dependent upon the thallus, that is the gametophyte, for the food material, water and mineral in solution. Thank you.